Hello guys, welcome to Rebuild Academy. My name is Adil Momin and I welcome you to the 10th part of our series From Virus to Vaccine, Let's Fight Infodemic. I hope you all are doing well, fighting pandemic as well as infodemic. But before we begin, let me tell you a very important point. It is extremely important and highly recommended to watch this series in order. Only then you will get a complete idea and a clear picture of every concept involved. The link is given in the description. Now, let's begin. In the previous discussion, we talked about medical treatment strategies in modern medicine. We specifically discussed preventive treatment methods for COVID-19, in which we talked about different aspects of COVID appropriate behavior, after which we learned about immunization, active and passive immunization. Later, we discovered how active immunity can be obtained by inoculation and how inoculation was done through variolation in the past. Then we discussed how the first vaccine was made by Dr. Edward Jenner for smallpox, and in the end, we learned the major steps involved in the manufacturing of vaccines. Right. Today we will start our discussion with different types of vaccine, how they are different from each other, and a few examples of them. After which we will talk about vaccine development and the approval process, the stages involved in vaccine development, and also the difference between standard and emergency approval. Moving forward, we will discuss the types and status of different COVID vaccine as of July 2021. Okay then, let's begin. Now, starting with our first topic, types of vaccine. Based on what part of the pathogen is used in vaccine making, there are three categories in which they are classified. The first category is whole pathogen vaccines. In these vaccines, the complete pathogen is used in vaccine making. There are three types of vaccines that fall under this category: live attenuated vaccine, inactivated vaccine, and viral vector vaccine. The next category is antigen-based vaccines. Subunit vaccine comes under this category. And the third category contained vaccines that are made from the genetic material of the pathogen. Nucleic acid vaccines come under this category. Let's learn about them one by one. Live attenuated vaccine, as the name suggests, is made from the weakened virus. When a person is inoculated with a live attenuated vaccine, develops the infection, but this infection can be mild or without symptoms. The advantage of this vaccine is that it enables a strong immune response and the immunity obtained is long-lasting. Thus, no booster shots is required. A booster shot is an extra administration of a vaccine after an earlier dose. This is intended to increase the immunity against that antigen back to protective levels after memory against those antigens has declined through time. Live attenuated vaccines are not suitable for people with weak immune system because they are at risk of full infection. Rubella and measles vaccines are examples of live attenuated vaccines. Another type of whole pathogen vaccine is inactivated vaccine. In this vaccine, a killed or modified pathogen is used. Since the virus is not active, it cannot cause infection. The immune response produced due to inactivated vaccine is comparatively less strong. Therefore, based on the requirement, a booster shot may be required later. But this vaccine is suitable even for people with a weak immune system because there is no danger of infection. An example of inactivated vaccine is polio and hepatitis A vaccine. The third type of whole pathogen vaccine is the viral vector vaccine. This is an interesting type. They take a modified version of a different virus known as viral vector with a genetic code of antigen of that virus of which we want to acquire immunity. We just use the other virus to deliver the genetic code, and even the other virus is modified such a way that it will not cause any infection. The advantage we get from this is the strong immune response and due to which there is no need for a booster shot in many cases. But there is a catch here. If the person has immunity against the viral vector, then the immune system may destroy the viral vector before it can deliver the genetic code. To overcome this, scientists use viral vector which have not infected humans till now. Ebola vaccine is an example of viral vector vaccine. Subunit vaccines are made from antigen parts of the pathogen. Since only the part of the pathogen is used and not the complete virus, therefore there is no chance of infection. But the immune response is less strong in case of subunit vaccines. To increase the effectiveness of the vaccine, adjuvants are used, and some subunit vaccines may require booster shots based on requirement. Influenza and hepatitis B vaccines are examples of subunit vaccines. The third category of vaccines is nucleic acid based vaccines. There are primarily two types, DNA vaccine and mRNA vaccine. The small m here indicates messenger RNA. First of all, it is important to note that nucleic acid vaccines are different from viral vector vaccines. In the case of a viral vector vaccine, we use a viral vector, that is a different modified virus, to transfer the genetic material. But in nucleic acid vaccine, we don't use viral vectors. Electroporation is used to transfer genetic material to cells in case of DNA vaccine. Electroporation is a physical transfection method that uses an electrical pulse to create temporary pores in cell membranes through which substances like nucleic acids can pass into cells, whereas lipid nanoparticles are used to safely and effectively transfer the genetic material in the case of RNA vaccines. DNA vaccines are stable at room temperature, whereas RNA vaccines require a special temperature range to be maintained. The genetic material has to be delivered inside the nucleus of the host cell to start working, but RNA vaccine doesn't require their genetic material delivered inside the nucleus, which makes them comparatively easy to deliver. The experiments have shown DNA vaccines induce a small immune response in humans, but research is going on and results seems promising. No DNA vaccine has been licensed to use on humans till now, but tests on animals have been largely conducted. DNA vaccine has been made to protect horses against the West Nile virus. RNA vaccines can produce strong immune responses and are found to be very effective. RNA vaccines have been used for rabies, influenza, and more recently for the Zika virus. The important point to note here is the advantage nucleic acid vaccines has over other types of vaccines is they don't require the complete part of the pathogen in their manufacturing. The genetic material can be rapidly and efficiently made in the laboratory, which cut down the time of manufacturing substantially, and they are suitable for modification according to variants of the virus. Basically, you are dealing with the software here rather than hardware. But the technology is new and yet to reach its true potential. Now let's talk about vaccine development and approval process. This process is long and complex. The approving authorities are different in different countries. For example, Food and Drug Administration, that is FDA, is responsible in the US. Central Drugs Standard Control Organization, that is CDSCO, is responsible in India. We will specifically discuss procedures adopted by FDA. There are many stages in the vaccine development and approval process. Let's see one by one. The first stage is known as pre-human trials. The first step in pre-human trials is the exploratory stage. In this stage, the scientists identify the antigen and isolate it. Then comes the preclinical stage. In this stage, the scientists test their vaccine on animals, especially those who have similarities to humans in terms of anatomy, physiology, and genetics. The mouse genome is very similar to human genome, making mouse genetic research particularly useful for the study of human diseases. Then these animals after vaccination are exposed to live pathogen to test their immunity. If they show immunity to disease, then it indicates the vaccine is working. This methodology is known as challenge model. When we challenge the pathogen to infect the vaccinated animal. After the studies on animals show positive results, the company files an investigational new drug, that is IND application, seeking human clinical trials. Based on preclinical data, FDA will do an assessment as to whether the product is reasonably safe for initial testing in humans, and if it satisfies with the results, it grants license for human trials. The majority of the vaccine candidates fail in this stage. The second stage is human trials, in which the first one is called phase 1 trials. 20 to 80 adult volunteers are selected for testing, and they are kept under close medical monitoring to observe the effects of vaccine. If the results on these human subjects turn out to be positive, then phase 2 trial starts. In phase 2, the sample size is increased to 400 to 500 adults. While selecting these adults, special attention is given to include those from risk groups like people with weak immune system, people with comorbidities, and also people are selected randomly from a particular geographic area so that majority of the variables are covered. Based on test data, the number of doses, the time between two doses, and the method of delivery of vaccine is tried to be found out. If the results of phase 2 trial are convincing enough, then begins the phase 3 trials. In phase 3 trials, the sample is further increased. 1 to 10,000 people are selected. The primary purpose of phase 3 trials is to find out any rare side effects of the vaccine and to do an efficacy test. We will look into the efficacy matter in the upcoming video. Then comes the approval stage. After phase 3 trial are completed, then the company applies for biologics license application to seek commercial use of the vaccine. FDA will inspect the data of the trials, and based on its assessment, it will approve the vaccine and provide approval for its use. There are two types of approval. One is standard approval and another is emergency approval, also known as Emergency Use Authorization (EUA). We will shortly look into the difference between these two approvals. But before this, let's see what is post-license monitoring, also known as phase 4 trials. This is a feedback mechanism. The company collects the data about people who are vaccinated with their vaccine and tries to find out if there is any side effect is reported. There is a system in the US called the Virus Adverse Effect Reporting System, which accepts reports of adverse events and reactions that occur following vaccination. Healthcare providers, vaccine manufacturers, and public can submit reports to the system. This is really helpful in identification of any possible side effects even after the vaccine is approved by the authority. Now let's discuss standard and emergency approval. It is important to note the company has to decide the type of approval it wants, standard or emergency, at the time of IND application itself. Now let's see what happens if the company applies for standard approval. First, it will complete all the human trials of phase 1, 2 and 3. Then based on evidence of the results, the FDA will grant the standard approval. Then it will start manufacturing the vaccine and then it will distribute. So this is the ideal process. Now imagine the condition of an emergency like a pandemic. What can you do to shorten the length of above process? Here comes the concept of emergency approval. What emergency approval does is, it gives permission for testing as well as manufacturing, so by the time the testing will be completed, there would be some amount of vaccine readily available to use. But this does not mean that if trial results are not satisfactory, then also these vaccines will be used. If the results after phase 3 trials don't show the effectiveness of the vaccine, then the license will not be issued. It is the company that is risking its resources here. So, once the company gets emergency authorization, it will readily start distributing and start ramping up its production capacities. Now you understand the difference between Standard and emergency approval. Emergency approval doesn't compromise on the quality or effectiveness of vaccines. It's just a time-saving exercise. Now let's discuss COVID vaccines. It is important to note that every country has their own approval mechanism, and vaccines approved by a particular country will be recognized by that country only. For example, only 4 vaccines are approved to use in India as of now: Covishield, Covaxin, Moderna, and Sputnik V. As of July 2021, only 9 vaccines are approved by World Health Organization, but different countries have approved different vaccines to be used for vaccinating their population. We will discuss vaccines currently used for vaccination by countries and also a few which are under trial. Now let's discuss vaccines by their types. No live attenuated vaccine has been approved by World Health Organization. Covivac is a live attenuated single-dose vaccine developed by the company Codagenix, but it is currently in phase 1 clinical trials. Next is inactivated type vaccine. Covaxin, developed by Bharat Biotech in collaboration with the Indian Council of Medical Research, is inactivated type vaccine. Another example is Coronavac vaccine, which is developed by the Chinese company Sinovac Biotech. VLA2001 is also an inactivated whole virus vaccine under clinical trials. Now talking about viral vector vaccines, then Oxford AstraZeneca, which is also called Covishield in India, is a viral vector vaccine. Sputnik V and Johnson & Johnson vaccines are also viral vector vaccines. And if we talk about subunit vaccines, then Epivac Corona and ZF2001 are subunit vaccines. Corbevax or BioE is a subunit vaccine developed by Indian biopharmaceutical firm Biological E Limited, Baylor College of Medicine in Houston, US, and American company Dynavax Technologies. It is under phase 3 trials in India currently. Another subunit vaccine is Novavax, developed by Novavax, an American biotechnology company, and the Coalition for Epidemic Preparedness Innovations, that is CEPI, and it is undergoing trials in India under the brand name Covovax. There is no DNA vaccine currently approved by any country. ZyCoV-D is a DNA-based COVID-19 vaccine being developed by Cadila Healthcare with support from Biotechnology Industry Research Assistance Council. It has recently conducted its trial in India and has applied for emergency use. Pfizer-BioNTech and Moderna are mRNA vaccines approved by WHO and widely used in the US. ARCO and CVNCO are also RNA vaccines under clinical trials. So, this sums up our discussion on major vaccine types. If you have more than one option of vaccine and you are confused, then I would recommend contacting your healthcare provider or family doctor and ask for advice rather than blindly going for any vaccine. It is equally important that you should not wait for your personal choice either, because maximum vaccine coverage is also essential. So this brings us to the end of today's discussion. Now, as usual, let's quickly revise what topics we discussed today. We started today with the type of vaccines, in which we discussed live attenuated vaccine, inactivated vaccine, viral vector vaccine, subunit vaccine, DNA, and RNA vaccine. Moving forward, we talked about different stages in vaccine development and approval process, pre-human and human trials. Then we also discussed standard and emergency licenses, after which we saw a few COVID vaccines currently used around the world according to their types, and many more are in developmental stages. So this concludes today's discussion. In the upcoming video, we will be discussing some of the crucial concepts like vaccine efficacy, vaccine side effects, and herd immunity. So make sure you subscribe the channel and press the bell icon for timely notifications. If you enjoyed and learned something from this video, then make sure that you like the video and share it with others so that we all work together and battle the infodemic. If you have any suggestion for me, then you can comment below. That's all for now. We will meet in the next video. Stay safe, stay connected.